Okay, so we are at a new topic, right? The topic of protein synthesis and mutations. In the previous topic, you have learned about the structure of the different RNA molecules, the tRNA, RNA, and mRNA. So in this topic, we will learn about their roles or their functions during protein synthesis. We will look at how the information stored in the DNA is used to make proteins in the process of transcription, and for the eukaryotes, the post-transcriptional modification, and then the translation. Okay, so after you have learned about the process of making proteins, we will learn about mutations, including gene mutation and chromosomal aberrations, and we will look at diseases involving such mutations, the Down syndrome, as well as sickle cell anemia. Okay, so to look at the process of protein synthesis, we need to look at what is a gene. So a gene is a specific DNA sequence that codes for a product. The product can be a protein or a polypeptide, or a particular RNA molecule, like a rRNA or a tRNA. Okay, so in a chromosome, right? A chromosome is made of a DNA molecule, right? Or a chromosome is made of a molecule of DNA. So the DNA is very long, right? So within the DNA, there are certain segments or certain sequences that code for a product. So in my example here, right, I have this sequence that codes for amylase, an enzyme, and another sequence here that codes for a tRNA. Right, so the gene that codes for a protein would be transcribed into a mRNA molecule, which is then translated into the protein amylase. Okay, but for the gene that is coding for a tRNA, right, it is transcribed into a tRNA. The tRNA does not go on to be translated. Right, so the mRNA goes on to be translated into a protein, but the tRNA remains as a tRNA. And of course, there are other genes that codes for rRNA as well, and these genes in a eukaryote are found in the nucleolus. So the rRNA is also not translated into a protein and remains as rRNA. Okay? So looking at the central dogma, right, which describes the flow of information, right, from the DNA to RNA to the protein or to the polypeptide. Okay, so the genetic information is stored in the form of DNA, right, in the form of the DNA sequence, and the DNA, right, is replicated, as you have learned in the previous lesson, and after replication, right, the daughter DNA molecules are passed down to the next generation, to the daughter cells, right, after cell division. All right, so the genetic information is passed down in that way, right, through DNA replication. So when the gene, right, inside the DNA molecule is expressed, then this genetic information is transcribed into a RNA, or specifically a mRNA, right, and that happens either in the nucleus or the nucleoid, right, depending on whether it's a eukaryote or prokaryote. Then this mRNA is then translated into a polypeptide sequence, which folds into a protein, and this happens in the cytoplasm, right, regardless of which kind of cells it is. Right, so this unidirectional flow of information at first, which is stored in the DNA, and when the gene is expressed, it is transcribed and translated, and then the information is flown from the DNA to the RNA to the protein in the form of a mRNA, mRNA sequence after transcription, and a amino sequence after translation. All right, so this is unidirectional. So by right, the information don't flow back, in the exception of viruses. Right, so there's a kind of virus called retrovirus that actually has RNA as its genome. Right, so we call the first ever lecture, we learn about cells, Then you learn about viruses, right? The first ever topic, right? So some viruses have RNA as a genome, and for this kind of virus known as retrovirus, right, the RNA genome is actually copied into DNA once the virus has infected the host cell. Right, so that's actually a reverse flow, right, from RNA to DNA. Okay, so this is an example of an exception, right, and an example of a retrovirus is actually HIV. So you actually learn this later lah, when you learn about viruses. Right? So for now, right, I just want to introduce to you an exception to the central dogma. Right, okay? So that would be the end of the first recording.